Chapter 4 Cell Structure 4.1 Studying cells. By the end of this section, you will be able to do the following. Describe the role of cells in organisms. Compare and contrast light microscopy and electron microscopy. Summarize cell theory. A cell is the smallest unit of a living thing. Whether comprised of one cell, like bacteria, or many cells, like a figure 4.1, a. Nasal sinus cells, viewed with a light microscope, b. Onion cells, viewed with a light microscope, and c. Vibrio tasmaniensis bacterial cells, seen through a scanning electron microscope, are from very different organisms, yet all share certain basic cell structure characteristics. Credit A. Modification of work by Ed Uthman, M.D. Credit B. Modification of work by Umberto Salvanin. Credit C. Modification of work by Anthony D'Onofrio, William H. Fowle, Eric J. Stewart, and Kim Lewis of the Lewis Lab at Northeastern University. Scale bar data from Matt Russell. Introduction. Close your eyes and picture a brick wall. What is the wall's basic building block? It is a single brick. Like a brick wall, cells are the building blocks that make up your body. Your body has many kinds of cells, each specialized for a specific purpose. Just as we use a variety of materials to build a home, the human body is constructed from many cell types. For example, epithelial cells protect the body's surface and cover the organs and body cavities within. Bone cells help to support and protect the body. Immune system cells fight invading bacteria. Additionally, blood and blood cells carry nutrients and oxygen throughout the body while removing carbon dioxide. Each of these cell types plays a vital role during the body's growth, development, and day-to-day maintenance. In spite of their enormous variety, however, cells from all organisms, even ones as diverse as bacteria, onion, and human, share certain fundamental characteristics. Chapter Outline 4.1 Studying Cells 4.2 Prokaryotic Cells 4.3 Eukaryotic Cells 4.4 The Endomembrane System and Proteins 4.5 The Cytoskeleton 4.6 Connections between cells and cellular activities Human. We call it an organism. Thus, cells are the basic building blocks of all organisms. Several cells of one kind that interconnect with each other and perform a shared function form tissues. These tissues combine to form an organ, your stomach, heart, or brain. And several organs comprise an organ system, such as the digestive system, circulatory system, or nervous system. Several systems that function together form an organism, like a human being. Here, we will examine the structure and function of cells. There are many types of cells, which scientists group into one of two broad categories: prokaryotic and eukaryotic. For example, we classify both animal and plant cells as eukaryotic cells, whereas, we classify bacterial cells as prokaryotic. Before discussing the criteria for determining whether a cell is prokaryotic or eukaryotic, we will first examine how biologists study cells. Microscopy cells vary in size. With few exceptions, we cannot see individual cells with the naked eye. So scientists use microscopes, micro equals small, scope equals to look at, to study them. A microscope is an instrument that magnifies an object. We photograph most cells with a microscope, so we can call these images micrographs. The optics of a microscope's lenses change the image orientation that the user sees. A specimen that is right side up and facing right on the microscope slide will appear upside down and facing left when one views through a microscope, and vice versa. Similarly, if one moves the slide left while looking through the microscope, it will appear to move right, and if one moves it down, it will seem to move up. This occurs because microscopes use two sets of lenses to magnify the image. Because of the manner by which light travels through the lenses, this two-lens system produces an inverted image. Binocular, or dissecting microscopes, work in a similar manner, but include an additional magnification system that makes the final image appear to be upright. Light microscopes to give you a sense of cell size. A typical human red blood cell is about 8 millionths of a meter or 8 micrometers, abbreviated as 8 micrometers, in diameter. A pinhead is about 2/1000ths of a meter, 2 millimeters, in diameter. That means about 250 red blood cells could fit on a pinhead. Most student microscopes are light microscopes, figure 4.2a. Visible light passes and bends through the lens system to enable the user to see the specimen. Light microscopes are advantageous for viewing living organisms, but since individual cells are generally transparent, their components are not distinguishable unless they are colored with special stains. Staining, however, usually kills the cells. Light microscopes that undergraduates commonly use in the laboratory magnify up to approximately 400 times. Two parameters that are important in microscopy are magnification and resolving power. Magnification is the process of enlarging an object in appearance. Resolving power is the microscope's ability to distinguish two adjacent structures as separate. The higher the resolution, the better the image's clarity and detail. When one uses oil immersion lenses to study small objects, magnification usually increases to 1000 times. In order to gain a better understanding of cellular structure and function, scientists typically use electron microscopes. 96 Chapter 4, Cell Structure Access for free at OpenStax.org. Figure 4.2. Most light microscopes in a college biology lab can magnify cells up to approximately 400 times and have a resolution of about 200 nanometers. B. Electron microscopes provide a much higher magnification, 100,000x, and have a resolution of 50 picometers. Credit A. Modification of work by GCG, Wikimedia Commons. Credit B. Modification of work by Evan Bench, Electron Microscopes. In contrast to light microscopes, electron microscopes, Figure 4.2b, use a beam of electrons instead of a beam of light. Not only does this allow for higher magnification and, thus, more detail, Figure 4.3, it also provides higher resolving power. The method to prepare the specimen for viewing with an electron microscope kills the specimen. Electrons have short wavelengths, shorter than photons, that move best in a vacuum, so we cannot view living cells with an electron microscope. In a scanning electron microscope, a beam of electrons moves back and forth across a cell's surface, creating details of cell surface characteristics. In a transmission electron microscope, the electron beam penetrates the cell and provides details of a cell's internal structures. As you might imagine, electron microscopes are significantly more bulky and expensive than light microscopes. Figure 4.3. These Salmonella bacteria appear as tiny purple dots when viewed with a light microscope. B. This scanning electron microscope micrograph shows Salmonella bacteria, in red, invading human cells, yellow. Even though subfigure, B, shows a different Salmonella specimen than subfigure, A, you can still observe the comparative increase in magnification and detail. Credit A. Modification of work by CDC, Armed Forces Institute of Pathology, Charles N. Farmer, Rocky Mountain Laboratories. Credit B. Modification of work by NIAID, NIH. Scale bar data from Matt Russell. Link to learning for another perspective on cell size. Try the How Big interactive at this site, http://openstax.org/l/cell_sizes. Cell theory. The microscopes we use today are far more complex than those that Dutch shopkeeper Antony van Leeuwenhoek used in the 1600s. Skilled in crafting lenses, Van Leeuwenhoek observed the movements of single-celled organisms, which he collectively termed animalcules. In the 1665 publication Micrographia, experimental scientist Robert Hooke coined the term cell for the box-like structures he observed when viewing cork tissue through a lens. In the 1670s, Van Leeuwenhoek discovered bacteria and protozoa. Later advances in lenses, microscope construction, and staining techniques enabled other scientists to see some components inside cells. 4.1. Studying cells. 97. By the late 1830s, botanist Matthias Schleiden and zoologist Theodor Schwann were studying tissues and proposed the unified cell theory, which states that one or more cells comprise all living things. The cell is the basic unit of life, and new cells arise from existing cells. Rudolf Virchow later made important contributions to this theory. Career connection. Cytotechnologist. Have you ever heard of a medical test called a pap smear? Figure 4.4. In this test, a doctor takes a small sample of cells from the patient's uterine cervix and sends it to a medical lab where a cytotechnologist stains the cells and examines them for any changes that could indicate cervical cancer or a microbial infection. Cytotechnologist, cyto equals cell, are professionals who study cells via microscopic examinations and other laboratory tests. They are trained to determine which cellular changes are within normal limits and which are abnormal. Their focus is not limited to cervical cells. They study cellular specimens that come from all organs. When they notice abnormalities, they consult a pathologist, a medical doctor who interprets and diagnoses changes that disease in body tissue and fluids cause. Cytotechnologists play a vital role in saving people's lives. When doctors discover abnormalities early, a patient's treatment can begin sooner, which usually increases the chances of a successful outcome. Figure 4.4. These uterine cervix cells, viewed through a light microscope, are from a pap smear. Normal cells are on the left. The cells on the right are infected with human papillomavirus, HPV. Notice that the infected cells are larger. Also, 2 of these cells each have 2 nuclei instead of 1, the normal number. Credit. Modification of work by Ed Uthman, MD. Scale bar data from Matt Russell.